Hi, Dr. Woeller here for Integrative Medicine Academy, and I welcome you to this particular webinar. This should be an interesting talk, and I talk about the fundamentals of oxidative stress and the neutralization of that oxidative stress via antioxidants. So some of this material actually comes from our Toxicity Mastery Course, which I'll discuss here shortly. If this is the first time hearing me, I have been an integrative and functional medicine physician since the late 90s. I do a tremendous amount of speaking, both online as well as around the country and internationally. I've written a number of books. I love clinical education. I'm also a practicing clinician. I do a lot of clinical education for Great Plains Laboratory, as well as Integrative Medicine Academy, which is our own online academy. We also have a website called Functional Medicine Clinical Rounds, which is a membership site for practitioners interested in more one-on-one input with regards to lab testing and case presentations, etc. And then in my own practice, I've been involved now for about 25 years with the autism community, patients with chronic and environmental health problems that could be chemicals, metals, mold, mycotoxins, etc. So when we think about free radicals, okay, so we've got some type of toxic exposure, and we've got various enzymes that are meant to help break those toxins down, primarily in the in the liver. There's always going to be an aspect of what's called free radical formation. Now, free radicals can also exist as oxygen radicals, which tax our antioxidant reserves, such as the superoxide dismutase or glutathione. And if we can properly convert oxygen radicals through hydrogen peroxide, we can reestablish levels of oxygen and water. But through that process, we also increase the potential of producing what are called hydroxyl radicals, which then can have a damaging effect against various cellular structures, particularly cell membranes, which cause lipid radical formation, lipid peroxidation, and then organelle damage within the cell, like the mitochondria. So we're going to talk about some of these things, and then the importance of various antioxidants and how they can neutralize against some of the toxic assaults that occur with these particular radicals. So first off, let's talk about oxidative stress and the reactions of oxygen. So oxidative stress is defined as a condition in which the generation of reactive oxygen species exceeds our body's ability to neutralize it and then protect ourselves against those particular toxins. So essentially, increased reactive oxygen species results in an increase in oxidative damage to various biomolecules in our body. Now, oxidative stress is a characteristic feature of inflammatory diseases. So if there's chronic inflammation, there's going to be oxidative reactions occurring. And then too much of it, of course, leads to cellular and tissue damage. And so various examples, right? Oxidative reactions in the joints can lead to arthritis, in the vascular system lead to atherosclerosis, or if it's just systemic, in things like systemic lupus or diabetes, all of that leads to various diseases. So it makes sense if we have good antioxidants or enzymes involved in antioxidant pathways, we're going to be protected. So glutathione, vitamin C, vitamin E, vitamin A, superoxide dismutase, catalase enzymes, glutathione peroxidase, all of those work to our advantage in decreasing oxidative reactions. And then again, anything from metals, chemicals, molds, drugs, smoking, alcohol, other things that can generate oxidative reactions, hyperglycemia chronically and diabetes, etc., will tip the scales in the other direction. So this is actually an interesting slide here. Let me grab a pen. And essentially what this is talking about is that the way oxygen is written in many textbooks is not how it typically exists. So the singlet oxygen, or O2, is not really how it exists in our body. Essentially, oxygen in our body as a ground state oxygen exists as a diradical at body temperature. And so from this point, it can form through different chemical reactions, either a superoxide ion or anion radical, or it will sort of equilibrate with this hydroperoxyl radical. So basically these two things are sort of fluxing back and forth, if you will. From a superoxide, we convert into hydrogen peroxide, and that has its own potential problems, but hydrogen peroxide is a normal product of development in our body with regards to movement of oxygen through superoxide, etc. And then the hydrogen peroxide gets converted into a hydroxyl radical, which is very toxic, or it can also form what's called a hydroxide anion base, and ultimately as well through water. So we need various enzymes to help us get out of this. So we have superoxide dismutase, we have catalase, and then we have glutathione that helps prevent against excess formation or accumulation, particularly of the hydroxyl radical, which is extremely toxic in biological systems. So molecular oxygen, which the O2, is essential for all aerobic life forms. So oxygen is relatively inert at body temperatures, but it does exist again as that diradical form. But at higher temperatures, it becomes highly reactive in combustion reactions. So with our body temperature fairly well regulated, we're not really at a baseline level producing a lot of oxygen radicals just on its own. That would actually occur through oxidative reactions that occur primarily in the cell for energy production. Now, approximately 90% of the consumed oxygen is linked to what's called oxidative phosphorylation, which takes place in the electron transport chain of the mitochondria for the production of adenosine triphosphate, or ATP. So if we think about aerobic respiration, well, anaerobic respiration involves glucose to pyruvic acid. Aerobic respiration involves what's happening in the mitochondria. So we get about 5% of our ATP production just through glycolysis. We get about another 5% through the Krebs cycle. But we get the bulk of our ATP produced through oxidative phosphorylation of the electron transport chain in the presence of oxygen. So it's all about getting to the level of acetyl coenzyme A in order to move things forward into the Krebs cycle, to move things forward from there into the electron transport chain to get the bulk of ATP. And so if you look at the mitochondria, we have the outer membrane and we have an inner membrane. We have an inner membrane space, which is where we get accumulation of hydrogen ions or protons that get pumped from the matrix into this inner membrane space. And that occurs through electron transport between the different complexes of the electron transport chain. So this is our electron transport chain that is transferring electrons. At the same time, we're getting an outflow of protons into that inner membrane space, so they accumulate over top of our ATP synthase, or what sometimes is called complex 5. And when we get that concentration gradient over top of the ATP synthase, it flows back into the matrix and forms ATP. This is also where the Krebs cycle exists. It actually exists in the matrix of the mitochondria. So if we zoom in a little bit closer into the electron transport chain, essentially the name tells us what's happening. We're transferring electrons along a chain reaction through these different complexes where we convert oxygen into water. At the same time, we're pumping out protons into that inner membrane space for them to accumulate over top the ATP synthase to influx back into the matrix, and that brings together the formation of ATP through the combination of adenosine diphosphate or inorganic phosphate that's sitting at the lower structure of this complex 5, or what's called the ATP synthase. So basically, we're synthesizing ATP through actions of this channel. That reaction is key. It is the conversion of oxygen or molecular oxygen to water with the combination of electrons that have been transferred along the electron transport chain. So oxygen utilization is critically important for oxidative phosphorylation, about 90%. Another 10% or so of oxygen is used by various enzyme systems in what are called hydroxylation and oxygenation reactions. So hydroxylation is adding a hydroxyl group or an OH group. And then another 1% of oxygen gets formed into reactive oxygen species, which is what we need to deal with metabolically to prevent oxidative stress. So an example of a hydroxylation reaction is the dopamine beta hydroxylase or monooxygenase in what converts dopamine to norepinephrine. So this is called dopamine beta monooxygenase, or what's also called dopamine beta hydroxylase. And so we're adding a hydroxyl group to the dopamine to make it norepinephrine. So that's a hydroxylation reaction utilizing molecular oxygen. From a fundamental aspect of just oxidative stress, okay, sometimes we talk about things that are reduced or things that are oxidized. And so this is just a kind of a basic concept. And that when we transfer an electron from one compound or one element to the next, okay, we're causing a reduction reaction. So essentially, when we transfer an electron to an oxidized compound, that oxidized compound becomes reduced. So reduction is the gain of an electron. Oxidation is the loss of an electron. So if we could just say over here, reduction is the gain, oxidation is the loss. So a reducing agent reduces an oxidized compound. And therefore, in the process, the reducing agent becomes oxidized because it's lost an electron. So reactive oxygen species are oxygen compounds that carry an unpaired electron or pairs of electrons. So common forms of reactive oxygen species that we produce naturally are superoxides. So this is actually an example of a superoxide radical or hydrogen peroxide, which can become problematic on its own. Now, it turns out that these things are also important metabolically. So reactive oxygen species are important in metabolism, such as certain enzymes. So we'll actually use hydrogen peroxide as a substrate. So if you think about this reaction here, if we form hydrogen peroxide, the way that we can neutralize and prevent that hydrogen peroxide from becoming problematic is converting it into water and oxygen. So essentially our body is recycling compounds for further use, like water and oxygen, to be used again in oxidative reactions. We need catalase in order to act upon hydrogen peroxide for the production of water and oxygen. So there are different types of reactive oxygen species that have an unpaired electron. So here we have molecular oxygen can become a superoxide anion. We can form peroxides. We can form a hydroxyl radical, which is very toxic. We can also form a hydroxyl ion. Okay, so it's these radicals or these unpaired electron species that are quite problematic. And from a very simplistic standpoint, if we look at any radical, okay, it's going to have an unpaired electron in its outer shell. And so let's say we take this sort of standard radical, it's going to want to grab an electron from something else. So in this case, it can grab it from DNA, and it will satisfy its outer shell, make it stable, but it leaves a wreckage of tissue in its wake. And so essentially that's what radicals are doing all throughout our body, whether it's DNA or RNA or a cell membrane or some protein. Essentially that is what's happening. So we have to watch out for overproduction of superoxides and certainly these hydroxyl radicals. Okay, so superoxide dismutase is an enzyme that helps to convert a superoxide anion into hydrogen peroxide. But hydrogen peroxide can form a hydroxyl radical. So we need catalase in order to convert it to water and oxygen, or we need glutathione to break that hydrogen peroxide into water. So our glutathione peroxidase and our catalase enzymes are critical in getting rid of a buildup of hydrogen peroxide. Again, these radicals can be damaging, particularly the hydroxyl radical. What's interesting about this when you start looking at things from an immunological standpoint. So reactive oxygen species are utilized by our immune system in a strategic way. So myeloperoxidase is an enzyme that gets produced by neutrophils. And that is used at a fundamental level in our tissue to destroy pathogens that have either invaded or are attempting to invade our tissues, like our gut, for example. So in this particular case, myeloperoxidase forms something called a hypochlorite ion, which then attacks candida. A better description of this is neutrophil myeloperoxidase forming this hypochlorous ion or hypochlorous acid to kill various pathogens. It's not just candida, it could be bacteria, etc. And so myeloperoxidase is an enzyme found in neutrophils. There are actually deficient states of myeloperoxidase. So people basically have an ineffective neutrophil system, which is one of the major components of our innate immune system. So NADPH oxidase, so nicotinamide adenine dinucleotide phosphate hydrogen, the oxidase uses molecular oxygen to form a superoxide anion that gets converted into hydrogen peroxide, and then the myeloperoxidase enzyme combines the hydrogen peroxide and a chloride ion to make hypochlorous acid. And that is what kills a pathogen at a tissue level. Now, if we think about how we can have a runaway effect of oxidative stress, a great example is just looking at lipid oxidation. So here is a typical description of a polyunsaturated fatty acid. The things that how we would come in contact with a polyunsaturated fatty acid would be from our diet, like vegetable oils, for example. So canola oil, safflower oil. It's one of the reasons that you want to try and avoid vegetable oils in your diet, because they carry multiple double bonds, which then get attacked by radicals. Okay, so here is our hydroxyl ion or radical attacking a polyunsaturated fatty acid. Now, vitamin E helps as an antioxidant in fat lipid peroxidation, as well as glutathione. If we work through the process of lipid oxidation of polyunsaturated fatty acids, eventually we'll form a couple compounds, malinaldehyde and 4-hydroxynonenal. I'll come back and talk a little bit about aldehydes here shortly, but notice that each of these compounds has this functional group called an aldehyde. And aldehydes by themselves are potentially problematic. So let's just kind of walk through this slide from A to B to C just to kind of get a general overview. Okay, so a hydroxyl radical attacks a polyunsaturated fatty acid. So it's going to form a carbon-centered what's called lipid radical. So here's our lipid radical right here, that little dot. The radical rearranges to form something called a dienyl radical. Okay, so basically it's just reorganizing itself, and that becomes a further propagating attack mechanism to attack further polyunsaturated fatty acids that are in this chain reaction. So now all of a sudden we form something called a hydroperoxyl radical, which then goes further and attacks down here or forms something else called a lipid peroxide. So this is if you ever heard the term lipid peroxidation, it's getting formed because of these self-propagating radicals that get initiated on a polyunsaturated fatty acid by a hydroxyl radical, which then backtracks to some type of oxidative reaction. Now, the slide basically ends at this R dot or this R radical. The R basically designates a chemical chain configuration. It's not designated because it depends on what kind of polyunsaturated fatty acid it is. So it's basically saying that there is this self-propagating fatty acid lipid radical that sort of loops around and goes back to the beginning and sort of starts this whole process again. And so this looping around process will occur until the supply of polyunsaturated fatty acids is exhausted or some type of antioxidant steps in and terminates the reaction. So vitamin E, for example, could step in and block the reaction at multiple sites. If the reaction continues on, we'll form the malondialdehyde or the 4-hydroxynonenal, okay, which themselves become toxic compounds. One of the things that these compounds do is they go off and attack proteins. And so they can form what are called advanced glycation end products. The most famous advanced glycation end products that we see in most clinical practice, clinical medicine, is hemoglobin A1C for diabetics. But there are many other types of advanced glycation end products that can occur. So basically the point is lipid peroxidation can end up leading to protein malformation within the cell. So if we think about the cell structure of a mitochondria, we have the outer membrane, we have the inner membrane, and all of these sort of protein structures in between, essentially. Okay, and so there's a lot happening at a mitochondrial level of where things can go wrong with excess oxidative reaction. So fundamentally, a mitochondrion is a double membrane organelle made up of phospholipids and proteins with 5 distinct parts. We have the outer mitochondrial membrane, the inner membrane space, which is where we accumulate the protons after they've been pumped out of the matrix, the inner mitochondrial membrane, things called cristae, which I'll show you here in a second. These are the infoldings of the inner mitochondrial membrane, and then the matrix, which is where our Krebs cycle exists. Now, the inner mitochondrial membrane is where a lot of the action is. This is where the oxidative phosphorylation takes place of the electron transport chain. So there are 3 main functions of the inner mitochondrial membrane. It's what houses the electron transport complexes that transfer electrons along that chain reaction. We produce the ATP through the ATP synthase. And the membrane is also important for transferring proteins which regulate metabolite flow into and out of the matrix of the mitochondria. Now, interestingly, the inner mitochondrial membrane has a protein to phospholipid ratio of about 3:1. But in the mitochondrial membrane is a specific type of phospholipid called cardiolipan. Now, cardiolipan is very important because of its chemical configuration. I'm going to show that to you here in a second. The inner mitochondrial membrane is folded into what are called cristae. And the cristae expand the surface area for ATP production. Now, in a mitochondria, or excuse me, in a cell, in an organ that is highly metabolic, like the liver or the brain, there's generally more mitochondria per cell, but there are more cristae folds in the mitochondria of those cells. And it makes sense because metabolically, we want as much surface area to make ATP in a highly metabolically demanding cell, in a highly metabolically demanding organ. So a key aspect of the inner mitochondrial membrane is cardiolipan. Now, cardiolipan gets its name in the fact that it was first identified in heart tissue of cattle. Now, it's found almost exclusively in the intermitochondrial membrane of plants and mammals. Its main function is to enhance function of the electron transport chain along the intermitochondrial membrane. So cardiolipin makes up about 20% of the total lipid composition of the intermitochondrial membrane. Now, most phospholipids that make up our cell membranes have 2 fatty acid chains. What's unique about the cardiolipin is it has 4 fatty acid chains. Okay, and so there's a reason for that. And the reason is that the cardiolipin, because of its chemical configuration and the way it orients itself into the intermitochondrial membrane, helps to compartmentalize and optimize function of the electron transport chain complex. So you'll notice that this particular image shows the complex from complex 1, 2, 3, 4, and 5 grouped together. So these complexes are grouped together along the cristae folds. And so each complex is producing its own ATP. It's not as though there's this, there's obviously, I don't know how many there are of these things, probably thousands, hundreds of thousands in a typical mitochondria, for example. Okay, so it's really interesting. So if we have a problem in our cardiolipin, that is going to create instability in our electron transport chain complex, which is going to compromise our ability to make ATP. Oops, let me see here, sorry. So there are many clinical implications for cardiolipin disruption. They've looked at it with regards to Parkinson's disease and Alzheimer's, just general aging, for example, disruption of various complexes, diabetes. Kind of makes sense now if we think about it from a diabetic standpoint, where we start creating lipid peroxidation, for example, that then can lead to advanced glycation end products, protein malformation, cancer. Okay, cardiolipin disruption can impair mitochondrial function and bioenergetics. This has been something that has been looked at with regards to cancer regulation within a cell. If we have faulty intermitochondrial membrane, okay, we're going to have faulty ATP production. We're also going to have increased oxidative stress, and increased oxidative stress leads to downregulation of various antioxidant systems and further damages the mitochondria in its ability to make ATP. Bartonella infections, chronic fatigue syndrome have been associated with problems of the cardiolipin. They think in Bartonella, for example, that certain forms are producing toxins or antibodies that attack the cardiolipin. I thought it was interesting that there are some estimates that upwards of 95% of people with debilitating chronic fatigue may likely have something wrong or something attacking their cardiolipin. Antiphospholipid syndrome, this is a syndrome where there are high amounts of anticardiolipin antibodies. And what it can do is it can lead to thrombosis, for example. And one of the things to be suspicious of is, let's say, a young woman who just has recurrent spontaneous abortion. So maybe some problem in their body forming thrombotic events or tissues that are causing issues. One of the other aspects of this discussion in just the general function of the mitochondria are the various enzymes that are involved in the Krebs cycle and the complexes of the electron transport chain. And this goes to the heart of runaway oxidative stress. Okay, so this enzyme right here called econotase. Econotase is an enzyme that's involved in the first stages of the Krebs cycle. It's what helps convert citric acid into isocitric acid. And embedded in that enzyme is something called an iron sulfur cluster. And that iron sulfur cluster has to go through a chemical switch or process in order for it to be functional to help the econotase enzyme as a complex function normally. Well, if we have excess superoxide formation, for example, okay, that can inhibit the proper configuration of iron. And if we form hydrogen peroxide, that produces a hydroxyl radical that can't get neutralized quick enough by intracellular glutathione. It's going to rob the glutathione reserves and then leave the oxygen radicals in a position where they can disable the iron sulfur cluster, which then basically disables the enzyme. Well, it turns out in the electron transport chain, those iron sulfur clusters also exist. So they exist in complex 1 and complex 2 and complex 3. Remember, these things are important along with CoQ10 in these complexes for the transfer of electrons and the outward flow of protons for the ultimate production of water in combination with oxygen. All of that leads to an accumulation of protons over the ATP synthase and the production of ATP. So it's really critical that these things are working appropriately. There's a lot of nutrition that's involved. In fact, they figure from a mitochondrial level, almost every nutrient that our body uses at a cellular level, the mitochondria are going to use. Vitamin A has a stabilizing effect on the intermitochondrial membrane. Copper is intimately involved as a regulator over different oxidation states of iron. Very interesting compound, which I'll talk about at some other point through Integrative Medicine Academy, and which I will actually bring up in our Mitochondrial Mastery Course, is methylene blue. Methylene blue is a very interesting compound, and what it does is it helps for the steady state supply of electrons, even if some of these individual complexes are disrupted. In fact, they use it in emergency rooms for carbon monoxide poisoning. So what it does is methylene blue essentially donates electrons, okay, combining with oxygen to form water to keep this process moving forward so that we can get some protons pumped into that intermembrane space for the production of ATP. So it's a really fascinating chemical. Now, all of this we talk about in great detail in our Toxicity Mastery Course. We go through aspects of chemicals, molds, mycotoxins, oxidative reactions, etc. We also get into, just from a pure mitochondrial standpoint, in great detail in our Mitochondria Mastery Course. So if you're interested in learning more about toxicity issues, check out the Toxicity Mastery Course, and we currently have a course going on now for Mitochondria Mastery, which is fascinating information. Let me talk a little bit about chemicals. So we start to think about chemicals' ability to generate oxidative reactions. So in my practice, I use a lot of the GPL Tox profile, which is the toxic non-metal chemical profile from Great Plains Lab. It looks at a wide variety of chemical compounds, from organophosphate pesticides to pyrethroid insecticides, etc. And one of the compounds that's pretty interesting on this test is something called acrylene. Now, acrylene is an environmental pollutant. It's commonly used as an herbicide. They also use it as an algicide, so they'll put it in like water aqueducts, for example. It could also be acquired through cigarette smoking, burning of gasoline or oil, and then certain bacteria like Clostridia bacteria could even produce some acrylene. One of the things I want you to pay attention to is notice that the acrylene has this functional side group here, a carbon double bonded to an oxygen with a hydrogen. That's called an aldehyde. So there's our aldehyde right here. Now, what's important about this discussion is that in organic chemistry, and I'm not an expert in organic chemistry, it's one of the more difficult chemistry classes in college, but what are called carbonyl groups. This is a functional group where a carbon is simply double bonded to an oxygen. Okay, and so there's different types of carbonyl groups: ketones, aldehydes, carboxylic acids, amides, etc. And this is not to insinuate somehow that all or every single one of these chemical forms in any particular compound is going to be a radical. But there are, the way electrons are moved and the aspects of organic chemistry can form radicals that can be damaging within our cellular tissues. And that particularly is very true of aldehydes. So here's acrylene, which is an aldehyde that gets acted upon by glutathione in attempts to render it less toxic. And so it formed a compound, called, glycinase and acetyltransfer. Excuse me, it basically converted it, and then we have another enzyme which basically tries to metabolize it. So in this particular process, acrylene is getting metabolized into a couple different compounds. So on the GPL tox profile, we're looking at a metabolite called 3-hydroxypropyl mercapturic acid. And so we have to have functional enzymes like aldehyde dehydrogenase or aldoketo reductase in order for this reaction to occur appropriately. But from a very simplistic standpoint, okay, acrylene toxicity, that through its reactive aldehyde group can be problematic. And they've looked at aldehyde toxicity in multiple sclerosis and Alzheimer's and cardiovascular disease and diabetes, autism, etc. So if we're forming a radical that is an oxygen radical, that oxygen radical can cause problems at the cellular level, and fundamentally within the mitochondria. Now, it could also affect the cell membrane, it could affect the nuclear membrane, it could attack the DNA and the RNA, and it could attack our mitochondria. If we think about liver detoxification, so we have phase 1, phase 2. Phase 1 is where we're converting a lipid-soluble compound into a water-soluble compound. And the early reactions involve oxidation reduction, hydrolysis, for example. But you'll notice that the P450 enzymes are situated within phase 1. And phase 1 is very much involved in the processing of aldehydes. Of course, we need robust nutrition, B vitamins, antioxidants, amino acids. And then eventually when we get over to phase 2, we're in a more water-soluble form, which can then excrete things either through the kidneys or the stool. This was a paper from a few years back that looked at aldehyde toxicity in general, whether it comes from alcohol, drugs, for example, or internal production. And so the basic idea is that many of these aldehydes, if not metabolized appropriately, can cause DNA and RNA damage, could generate reactive oxygen species, which we already know can cause lipid peroxidation. Lipid peroxidation byproducts can cause protein damage, advanced glycation end products. If we're causing lipid peroxidation, that could damage our mitochondrial membranes or just the membrane itself within the cell. If we have runaway oxidation, we're going to eventually have decreased glutathione, which is then just going to lead to further cellular problems. So when you look at this paper and dive in a little bit deeper, one of the things they talk a lot about is acetaldehyde. Now, acetaldehyde is the main product, if you will, of ethanol metabolism. So as our body tries to metabolize ethanol, it converts it into acetaldehyde. And we have to have acetaldehyde enzymes in order to convert acetaldehyde into acetic acid. Another thing that creates acetaldehyde in the body is candida. So a fungal problem in the gut or gastrointestinal candidiasis, if it's really significant, could be another factor of leading to acetaldehyde formation. I mentioned before that certain gut microbes particularly Clostridium type bacteria, can also produce acrylene. And again, acrylene has that aldehyde side chain or group that can be involved in oxidative reactions. So acrylene in an acute exposure high dose could lead to immune suppression, but in a chronic low dose over time, it might actually lead to just sort of chronic inflammatory responses. Now, if you think about it from an initial standpoint, if the exposure was really acute, it might just be a rapid ramp up and there might be some neutralization aspects of it. But usually people who are getting exposed chronically over time environmentally, it can have an accumulating aspect and create problems of chronic inflammation and eventual tissue injury. Now, one of the other things to look at, and we get into this quite extensively in the Toxicity Mastery Course, is mycotoxins. So there's all kinds of different mycotoxins, from the aspergillus mycotoxins of aflatoxin, ochratoxin, for example, gliotoxin. I want to take a look at Riordan E and Verrucarin A, because this doesn't get discussed all that often. These particular mycotoxins are called Trichothesanes. And Trichothesanes are a large family of fungal metabolites, okay, that come from things like Fusarium mold or Stachybotrys mold. Stachybotrys being that black mold, very dangerous, found in damp indoor environments. Fusarium can also be an environmental mold, but it can also be in food, grain products, for example. What's interesting about this discussion in the context of what we're talking about here as far as oxidative reactions comes down to characteristics within the chemical structure of a trichothecene. Now, these two trichothesanes are called macrocyclic, and that macrocyclic structure is this sort of expanded structure right here. Okay, so that adds some further chemical characteristics. But the thing that I want to focus on is right here, this triangular shape chemical structure where we have a bond to an oxygen in what's called an epoxide. So you'll notice that Riordan, Verrucarin, saprotoxin are these macrocyclic trichothesanes. But whether it's a macrocyclic or a non-macrocyclic trichothesanes, they all have this epoxide ring. Now, this epoxide ring is very unstable, or I should say it's highly reactive. Most epoxides are toxic because they're highly reactive in biological systems. So they're mutagenic, essentially. In fact, if you have high epoxide infiltration into the nucleus of the cell, it can impair the guanosine-cytosine pairing of the nuclear base. And that can then create problems from a protein formation standpoint off of the DNA template. So basically will cause sort of faulty RNA messaging. Now, we all in our bodies have what are called nucleophiles. Or a nucleophile essentially is a nucleus-loving compound. So nucleophiles provide electrons for creating a new chemical bond. So for example, a thiol is a nucleophile. So an example of a thiol would be glutathione. So glutathione has sulfur compounds that act as a nucleophile, meaning they donate an electron to provide a new chemical bond. In the context of a toxin, a thiol from glutathione will attack the epoxide triangular structure here, open it up, and then render that particular toxin neutralized or less toxic. So when we break it back fundamentally to the liver for detoxification purposes, we have these normal oxidation reduction reactions. We can add water, we can add oxygen, we can add hydroxyl groups, sulfur groups. But glutathione conjugation is huge. It plays a role in liver detoxification, particularly in phase 2. But this also shows us why we need robust nutrients in our diet. So thiols, which are naturally found in things like garlic, onions, other cruciferous vegetables, play a huge role. Sulforaphane, for example, from broccoli, can be important in some of this discussion. So there are various types of oxidative reactions. We talked a little bit about superoxide and hydrogen peroxide and the hydroxyl radical, which is very damaging. There's nitrogen radicals. We didn't really get into the peroxynitrite, for example, which could be quite problematic as well. So there's obviously more to discuss and get into here. We did talk about hypochlorous acid a little bit with regards to a neutrophil's ability to produce it to attack some type of pathogen like candida or bacteria. So there could be natural production in our body just because of metabolism, and then aspects of our immune system that produce it locally for its own particular use. But all of it fundamentally comes down to and gets balanced out by antioxidants. And so this brings us back to this concept of oxidation reduction and what's called redox, or redox. So again, a reducing agent will donate an electron, okay, to something that's oxidized for that oxidized compound or element to become reduced. So oxidation is the loss of an electron. Reduction is the gain of an electron. And so if we bring this back to this basic template of a free radical that has an unsatisfied outer shell, it wants to fill that gap with an electron, and so it's going to grab it from something. A superoxide anion will grab it from another chemical, another structure. It could be a lipid, could be a protein, could be DNA. Same thing with a peroxide, same thing with a hydroxyl radical. So it satisfies its outer shell, but it leaves damaged tissue in its wake. So that's an oxidation, a very simplistic way of looking at an oxidation reaction. So essentially an antioxidant helps to step in by donating an electron to the radical before the radical will grab an electron from something else, from a lipid, from a phospholipid, from a protein, from a nucleotide, whatever. And so that's what antioxidants are doing, is they're intervening before the radical has a chance to do tissue damage. And we think about fundamentally from a nutritional standpoint, why many of the antioxidants we get in our diet are so important, or things from a supplemental standpoint. And this is a whole other discussion that could be opened up, is the interplay between different antioxidants, the interplay between glutathione lipoic acid to vitamin C to vitamin E. And so there's a relationship chemically between these different antioxidants, moving from their oxidized to the reduced state, so what they are fully active and available to be involved in various chemical reactions for neutralization of oxidative stress. Ascorbic acid is particularly well suited for this in its various forms that it moves through. So a vitamin C exists as an anion at different physiological pHs. And so it helps to reduce a superoxide, for example, and is also involved in the reformation of vitamin E from after it has interacted, for example, let's say with a polyunsaturated fatty acid attack. It's also involved in the recycling, ascorbic acid, of glutathione. So again, there's this interplay between these antioxidants that's critically important from an antioxidant standpoint. Now it's interesting, there's other minerals that can be important in stabilization of cell function. Magnesium is not considered an antioxidant per se. Magnesium is really important in the cell, particularly in the mitochondria, because what it does is helps to stabilize ATP that gets produced through the ATP synthase in the matrix of the mitochondria. So it forms a chemical bond, stabilizing the ATP from it breaking down or reacting chemically prematurely, because then that ATP needs to be either used within the mitochondria itself or elsewhere in the cell. So there is an aspect of magnesium from an intracellular standpoint that's important, but it's not typically acting as an antioxidant. But what it does do sort of directly, it maybe has an influence on oxidative stress, is that magnesium regulates calcium. And calcium is a trigger of oxidative reactions in the cell. So one of the things that happens is when we get an increased flux of calcium into a cell, it can trigger different nitrogen oxygen species. And we can see here we're forming a superoxide, for example. We form a peroxynitrate radical that forms a hydroxyl radical. And then that can go off and attack proteins, attack cell membranes, attack the mitochondrial membrane. Excess calcium can create imbalances in the mitochondria that eventually lead to mitochondrial dysfunction. If we create problems or imbalances of intracellular calcium and magnesium, that can damage the mitochondria, and we get an outward expression of cytochrome C that moves into that intermembrane space and then activates what are called caspases that lead to cell death or apoptosis. Calcium can trigger lipases, for example, that break down the cell membrane, leading to an expression of arachidonic acid or a production of arachidonic acid. Well, arachidonic acid can generate its own oxygen radicals. So these reactive oxygen species can move outside the cell, and if this was happening in the brain, for example, it could damage glial cells or activate microglia, for example, for chronic inflammation. So essentially the way you could think of this is that magnesium, which is critically involved in the stabilization of mitochondrial activity, particularly with regards to stabilizing ATP, is also a regulator of intracellular calcium. So that's the information here for this particular lecture. I hope you enjoyed that and got some useful information. Some of the resources that I used for this particular lecture, fantastic article I would encourage you all to read called Melatonin in Mitochondria, Mitigating Clear and Present Danger. I think it's always worthwhile to have a good medical biochemistry textbook. I have a few. For this particular lecture, I used one from Elsevier Publishing called Medical Biochemistry, 5th edition. Magnesium Reversing Disease is a good book by Dr. Levy. And then I pulled content from our Toxicity Mastery course. I actually pulled content too from our current Mitochondria Mastery course. Both of these are available through Integrative Medicine Academy. And then there's other slides in here that are linked to specific research articles. If you're interested for more information about the Toxicity Mastery course, you can go to toxicitymasterycourse.com. And then if you have any questions about any of our courses, you can always check out our main website at integrativemedicineacademy.com, or feel free to email us with questions to integrativemedicineacademy@gmail.com. Also, if you look in the chat window, there is a link to a book, Toxicity book, as a complimentary download. There's a link there if you didn't see it already. I meant to mention it earlier, but you can click on that. That will take you to the website to get that book, just some information about toxicity, more information about the Toxicity Mastery course as well. And again, as I mentioned, you can always reach out to us with ongoing questions. Okay, everybody, thanks so much for your attention. I hope you found that interesting. I'm Dr. Kurt Woeller for Integrative Medicine Academy. Take care.